April 30th through May 3rd, we're going to have the 12th annual meeting for the Organization for Studies on Sex Differences. This is a great preclinical, clinical group of investigators who are with a common interest of looking at sex differences in a variety of different, preclinical and clinical conditions. And it'll be held at Lowe's Atlanta Hotel, beautiful hotel in the heart of Atlanta. And I'm the local organizing host, so please feel free to contact me or take a picture of the slide. We hope to have some flyers to advertise this as well. We're currently soliciting proposals for symposia, and the deadline's October 30th, but if it comes in a day or two late, you can just say, Ann said it was okay, and you'll be good to go. I know the chair of the scientific program. Actually, I'm on scientific program too, so you'll be good to go. And shortly thereafter, there will be a call for posters and individual talks. So we'd love to have as many people from this audience come and join us. And please also consider becoming a member. We're a brand new society, and the more members, the bigger voice we're going to have ultimately. So thank you.
Thanks, Ann. And I'd like also just point out that on the NIH campus on December 18th and 19th, there will be a free workshop about understanding traumatic brain injury in women. And so if you have any other questions about this, please reach out to Catherine Snedeker up here. She could provide some additional information if you're interested in that event. So with that, I'd like to invite our moderator for session 3 up to the podium. It's Dr. Colin Fletcher. He is the program director for the National Human Genome Research Institute, and he also plays a vital role in the knockout mouse phenotyping program, which is connected to the Common Fund. So please join me in welcoming Dr. Fletcher to the stand.
All right, good afternoon. This session is entitled Sex Differences in Animal Models, and I think we're going to be continuing with the same themes of sex, the brain, genes, the microbiome, the environment, all those topics. Our first speaker is Anna Penn, who comes to us from the Children's National Medical Center in Washington, and her talk is Sex and Perinatal Brain Damage. Why can't boys be more like girls?
I wish I could answer that question, but we'll at least show you where we've gone with looking at these issues. So I want to thank the organizers. This has been great. Most of the work I'm going to present today was funded by NIH, first by a New Innovator Award, and now by an R01 from NICHD. And we draw significant support from the IDDRC that is held by Children's National. So this is the question that when I started my independent research, I started with, why can't boys be more like girls? And that's because I'm a neonatologist. This is actually one of my patients. He was born at a very low birth weight, very early gestational age. You can see he's awfully cute. He's actually, this is a very old slide. I think he's probably about 10 years old now. But you can see that he does have significant neurologic damage. He has cerebral palsy. What you can't see is that he also has verbal communication difficulties and executive function problems. Okay. So, as we've said, there are significant sex differences between males and females that seem to start very early in development. And today I'm going to talk to you about adding another layer of complexity and looking even earlier in development during the in utero period at what may underlie some of these differences. But this is one of those slides that it's a little bit older now, but the data has held true. Males are always more susceptible to significant perinatal brain injury than females taken as a group. At 23 weeks, 24 weeks, and 25 weeks gestation, you can see that essentially males have a cognitive score on average that is about a week behind where a female would be. And this is, in fact, not true when you get to the term equivalent group. And so this puts many infants at risk, not just males, females as well, and we're interested in improving the outcome of both. But the question started with what the difference was between the male and female experience. And So this is just a schematic of early brain development, what happens in utero. Term is 40 weeks. The earliest edge of viability now has shifted, but is probably around 22 weeks. And this is the normal environment that the developing fetus would be in, with a nice big placenta there. And this is the environment that our NICU babies unfortunately find themselves in. And what we're interested in doing clinically is improving outcomes by mimicking this type of environment as best we can. And one way to do that is by looking at the placenta. And so this is my one claim to fame. I coined the word neuroplacentology and got Scientific American to publish it. So if you Google it, it is a word. It comes up. You know, everything that's in Google is true. So, so this is the idea that the placenta really plays a fundamental role in shaping fetal brain development, that this doesn't happen in a vacuum. This doesn't happen only after birth, but rather it contributes greatly to the organization and the health of the brain. And if we can understand what factors evolution has decided are important for neurodevelopment, perhaps we can improve outcomes by repleting them when a baby is born preterm, or if they just have placental compromise, so infection, hypoxia, any form of placental failure. So there are many components of this area, and groups actively working in many of them. Oxygenation and perfusion is what most people think of, growth and nutrition, stress regulation, immune activation, and we've already heard a little bit about immune activation in the developing brain and the sex differences that exist there. My laboratory is focused on hormones and hormone production from the placenta, and I'll tell you a little bit about that work today. So here's the question that we posed, and here is just a point-by-point highlight of, I think, what the key issues are. It's a multi-component system. We're talking about fetal sex playing a role in response to insults, but we're also talking about placental responses. Dr. Sadowski pointed out after the first talk this morning. It's always very striking that when I first started working in placental biology, if you go to look for the sex of a placenta in the pathology department, they're all female because they're all under the mother's name. So you actually have to read the path report to figure out whether it was a male or a female fetus. And everything stems from that. People think of the placenta as part of the mother. Genetically speaking, it is a fetal organ, and it's probably critical in the sex-specific responses that we see. This session was called Animal Models, so I'm going to try and focus mainly on the animal models that we have been using. To date, animal models have been quite limited. NIH now has a very active human placenta project, which is focusing on tools for use for analysis of particularly early human placenta, based on the idea, in part, that animal models have been insufficient. I think animal models are insufficient, but of utility in trying to tease apart some of these mechanisms. The Bale Lab, which has just moved to Maryland, has given us a couple of really nice examples of this. So if this is an area you're interested in, I highly recommend taking a look at their work. Looking at sex differences in nutritional transport, stress programming, in altering specifically male behavior, and altering insulin response, which alters gene expression in male placenta only, and therefore may set up this paradigm of males being more susceptible to in utero stressful events. And the reason we need to understand this is that to provide optimal treatment for the fetus and for the neonate, and potentially for the mother, we need to know what these mechanisms might be. So this is the maternal placental fetal unit, and the way I think about it in terms of programming later development, particularly developmentally programmed adult phenotypes, and what I'm specifically interested in is the neurologic outcome. This is the section of the unit that we think about in terms of sex-specific differences. And in terms of sex as a biological variable, I just wanted to point out we've already acknowledged that the placenta has the same genotype as the fetus. And I'm sorry, Peg, for using dimorphic. Sexually differential gene expression is seen both in the placenta and the brain. We've heard about some of the epigenetic markers that can be altered. It's worth noting that the epigenetics of the placenta and the brain may be quite distinct, even in the same organism. And so that adds another layer of complexity. Sex steroid and production also occurs both in the placenta and the fetus. And I'll tell you a bit about the placenta in a moment. And developmental injury is really highly dependent on the critical period during which an insult occurs. And that can be the critical period for the placenta or the critical period for the fetus and for the fetal brain in this particular case. So how do we use animal models to study neuroplacentology? Well, one of the questions is always how closely does form need to follow function for a model to be a useful tool? You can see the mouse doesn't have gyri. It does have a discoid placenta, much like the human placenta. On the other hand, the mouse placenta has a very different conformation. It doesn't invade the maternal uterus the way the human placenta does. It does have the same cell types as indicated by the different colors of the cells in the different layers, but you can see they're really quite dramatically rearranged in the two. On the other hand, there have been a number of nice studies looking at co-expression of orthologous genes in both species in the placenta, and over 80% of the genes that are known to cause placental phenotypes in mouse are co-expressed in both species, and many of them do correlate with human disease. So it's an imperfect model, but a model nonetheless. And we need this model to start to untangle what's going on between placenta and brain. So this is the system that we work in. We work in mouse, embryo, and postnatal mouse. You can see that mice lose their placenta in what we would consider late mid-gestation, whereas humans doesn't happen normally until up here. We use two different types of models. We look both at inflammation and hypoxia models, and I'll tell you a little bit about the hypoxia model to mimic preterm brain injury so that we can begin to look at the different hormones that may impact neurodevelopment. And then more recently, we've really focused all of our efforts on manipulating placental hormones themselves one by one to try and get a sense of what they're doing in the normally developing brain and then what we can do in terms of replacing them if they're compromised. So this is where we started. We started with a chronic hypoxia model of preterm brain injury, and it's quite a simple model where mouse pups soon after birth are exposed to 10% oxygen for 8 days and compared to normoxic group for the longest time. Of course, this was only done in male animals. We do it in a mixed model, and we sex balance our groups. And then we can either look at them immediately or evaluate their later outcomes. You see many similarities between what happens in these brains in terms of loss of cerebral volume, ventriculomegaly, white matter loss, which is quite significant in our preterm population, and impaired synaptogenesis. But we went on to look and see what would happen if we compared males and females who were exposed to this hypoxia. And we knew that both males and females had regional volume loss. It turned out when we actually looked volumetrically at the hippocampus, for example, in mice who had just been exposed to hypoxia, so this is during the period that's mimicking premature development and then sort of the perinatal term period, what we saw was that hypoxic male and female hippocampus were equal in size. So you might not have noticed there was actually a sex difference going on. But in fact, the male hippocampus is normally about 10% larger than the female. And so if you have an increase in volume loss, that evens out. But that is actually quite abnormal. We also saw myelin loss, notably in the male animals. And in the adults, what we saw was that the regional brain volumes in females recovered. Actually quite well, but the male hippocampal and cerebellar volumes remained reduced. And this correlated with adult behaviors in these animals where males who do not want to be put in a Morris water maze, which is a water-based test of mouse hippocampal function, they want to swim out as quickly as possible. These mice, the males, had a significant difference between how they did if they had been exposed to hypoxia, and females did not. And we've gone on to add to this model. We actually have now an even better model of preterm brain injury that combines the inflammation that we often see preterm with the hypoxia that can result postnatally in our preterm infants. But I promised I'd tell you more about neuroplacentology, so we're going to switch gears a little bit and talk about manipulating placental hormones. So the placenta is a neuroendocrine organ. It's actually the first endocrine organ to develop, and we've become very interested in this neurosteroid, allopregnanolone, that I'll tell you about. There are many, many hormones, though, on our short list to be studied, and our long list is about 104. So if anybody doesn't have anything to do, please come join us. So why neurosteroids? Well, we started thinking about why preterm infants might have susceptibility to neurologic damage, both males and females. And this is just weeks of gestation, maternal levels of progesterone and estrogen, and then the comparative fetal levels. And these infants who are born in the extremely preterm period are missing this enormous surge of exposure to what we think of as sex steroids, although here they may or may not be working in a sex-specific manner. So that's one of the things we wanted to figure out. So you can't target progesterone during a pregnancy, you lose the pregnancy. That's the end of the experiment. So instead, we started thinking about what the downstream derivatives of progesterone might be. And the one that's most interesting is allopregnanolone, which is a highly neuroactive steroid. It's made through a two-step conversion process, and it has this 3-alpha-hydroxysteroid dehydrogenase enzyme that's quite specific for its production. If most of you think of those horrible steroid pathways where there are things going off in all directions and their reuse of lots of enzymes, 3-alpha-HSD is fairly specific to this reaction, making it a relatively controlled experiment where we can manipulate the enzyme and change the allopregnanolone level. The other interesting thing about allopregnanolone is it doesn't work through the genomic mechanisms that you've seen discussed in terms of steroid hormones. Instead, it's non-genomic, and it primarily acts at extrasynaptic GABA-A receptor sites. And it plays a role in controlling tonic inhibition. GABA during cortical development, as was alluded to by Peg briefly when she was talking about the hippocampus, GABA plays many roles in neurodevelopment, and the striking thing is that it's not an inhibitory transmitter, at least very early in development. It's actually a synaptic depolarizing transmitter, but it also plays a role in almost every part of neural development. I'm going to summarize very quickly, for the sake of time, the key things that we've demonstrated in the lab recently that hopefully will be in publication soon. The placenta is the main source of allo during late gestation. It is made in the brain later on postnatally, but we now have evidence both from mouse and just this week also from humans that it is made by the placenta, and it peaks around 34 weeks of gestation and the equivalent in mouse. So we've looked by mass spectroscopy, we've looked at gene expression. Allo is easily transferred from placenta to fetal to the brain to the brain, so it crosses the blood-brain barrier. That's something that you would want to know, and we've done measurements in the brain as well using mass spectroscopy. Allo acts on GABA-A receptors, as I said, to modulate excitatory inhibitory tone, and we can show that we are altering this if we alter allopregnanolone levels in the fetus. To look at this, we designed a Crelox mouse. Most of you know, but I thought I'd just show it here. So the way Crelox system works, you clip out a specific gene when Cre is being expressed. If your gene happens to be surrounded by LOXP sites, we designed one that would knock out this particular enzyme. We were given the generous gift of a mouse from Wetzel and Lyon, in which the placenta expresses Cre. It's not expressed in the rest of the fetus. And so we could target 3-alpha HSD. We went to great pains to show that this is a valid model, that we're only getting changes in gene expression in this enzyme in the placenta, not in the brain, and that the steroid levels alter in the way that we expect. We get about 50% reduction in production of allopregnanolone in those placentas, and about a 50% reduction in the brain of those fetuses in which it has been reduced. So this is the setup. This would be the floxed mouse, and then the Cre mouse. It's actually expressing Cre under the control of a CYP19 promoter, which is the aromatase gene for those aficionados, because mouse placenta do not express aromatase. This is the human enhancer. And we can make litters that are either 25% control, 50% het, and 25% knockout, or 50-50, depending on what kind of breeding strategy we use. And so we're altering the placenta and then looking at the outcome. And this summarizes a tremendous amount of work by a very talented postdoc in my lab, Dana Bachler, who has been looking at what happens to the embryonic cortex once allopregnanolone is removed from the placenta. So this is just to show you, so this is through the cortex, from the top of the brain to the bottom of the brain, a wedge. We always are looking at somatosensory cortex in the same area in all of our mice, in embryos. She's labeled dividing cells with BRDU in the period when the layers are forming up here in the upper cortex, and then doing analysis 2 days later using markers that mark the upper layers in red, lower layers in green. And what you can see is that there are many more cells that have divided and gone to the upper layers in the normal animal than in the one that had low progesterone. The striking thing is that this loss of upper layer neurons, when you go back and break the code on these mice, it's significant only in the males. And we're seeing that persistently into adulthood, that it's really the upper layer neurons in males that create this difference. We've looked in adults as well at, there I go again, dimorphism. It's not, it's difference. A differential gene expression in the brain when we compare at P30 and do RNA-seq on different brain regions. So this is long after the insult has occurred, long after the manipulation has occurred. This has long-term consequences in terms of gene expression in cortex, hippocampus, and cerebellum. If you look at the males, you see many, many genes in the cerebellum, for example. I'm almost done. And not so many in the females. What's really striking, though, is if you look at the top diseases and biological functions here, in males, the top hits are almost all in myelination, and then in activation of microglia. I thought that might intrigue Dr. McCarthy, who's nodding. In females, it's almost all neurogenesis. So there's really a striking difference, and we're in the process of teasing this apart. We also see behavioral changes using standardized testing between male and female, not surprisingly. Untangling that has been a bit of a challenge, but, you know. And then the question is whether allo matters in human infants as well, because ultimately that's what I care about in terms of patient therapy. And we're looking both at human placenta and in the NICU. We're using sex as a variable for all of these studies, but I can't tell you yet how that's going to play out in the human population. But given the strong sex specificity of neurologic damage in preemies, I think it's a good bet. So one of the critical questions is whether the underlying mechanism of this susceptibility really is different between males and females, so that we're seeing changes in myelination in one and neurogenesis in the other, or is it simply the developmental timing of the events? Because one would imply that you need two different treatments. The other just implies that you need a treatment that's given at a different time. And the jury is still out. What we want to know is whether the placental hormones that we've identified can be used to protect the developing brain. First, we have to know what the timing is, and we're starting to do those experiments in humans to see if we can get some measures. And then, just in closing, just want to point, so sex differences exist in placenta, brain, and in the fetal placental unit as a whole. Placental endocrine function can specifically shape long-term sex differences. Placental hormones may provide new therapies for perinatal brain injury, and the preclinical models that we have are beginning to provide some mechanistic insights into these sex differences. So with that, I want to thank my research team, collaborators, and people who have worked in the lab previously, funders, of course, NICHD and the Office of the NIH Director, as well as many of our other funders. And this is my lab, and you'll note a little bit of sex skewing in my lab right now. We happen to be an all-female lab. I've never had that happen before. And in fact, I just accepted a faculty member who is male. I do not discriminate, but it's been a pleasure to work with a very strong and successful female cohort for the last number of years. Thank you.
I think we're gonna have to continue because we're a little behind already. You're gonna switch this over to.
Oh, yeah, of course.
So, with no further ado, Jane Danska from the Hospital for Sick Children in Toronto.
Thank you, Colin, and thanks for the invitation to come and join you today. I know that it's a broad audience, and I was given a variety of all important objectives for this meeting. I don't know if I'll meet all of them, but I'll try to not make it too much of a data-heavy discussion today. So I'm an immunologist, and I'm coming at this problem of sex differences and gender differences from the perspective of diseases that are immune-mediated, particularly autoimmune diseases. And as you likely know, there is a massive excess of female over male cases of many different autoimmune diseases, shown here with some abbreviations, males in blue and females in green. So this is prevalence, representation of a variety of different diseases, and they're ordinated from the most greatest bias in things like Sjogren's and systemic lupus down to RA, MS, and type 1 diabetes, about which I'll speak further. So this is a massive epidemiological footprint. For example, with the enormous amount of genomics that's been done to try to ascertain genetic variations that are associated with risk for autoimmune diseases, for example, in systemic lupus, the relative risk for lupus of having inherited all of the defined risk alleles that are known so far is dwarfed by the relative risk of being a female. But we have no idea why that's true. So that's, I think, for many of us, a bit of an elephant in the room. Very nice work from Carol Ober's group at the University of Chicago, who's been interested in sex effects in genetics for many, many years. Shows here that this female bias increases with age at onset of a variety of these diseases. And again, they're abbreviated here. That matters not, except that you see that for many of these diseases, the excess prevalence of female cases increases as the age of onset gets older and older. So one of the points that I wanted to make in favor of understanding mechanisms here is not just for the prevention and treatment of these diseases in women, but also to recognize that there is a richer, layered sets of information here. And I think it goes back to the important concept raised earlier about sex effects on as modifiers of disease and modifiers of gene activity. For example, in multiple sclerosis, although the number of cases is greater in females than in males, the difficulties of managing men with multiple sclerosis can often be far more challenging, and a higher frequency of males move into progressive MS, which is far more difficult to treat. So there are important issues here to the health and management of both male and female persons with autoimmune disease. I put the arrow up for type 1 diabetes, a disease on which we've worked for many years because it's the exception rather than the rule, in that it shows relative parity with regard to numbers of cases, remembering that diabetes is largely a disease of pediatric onset, and many of the cases present before puberty, and we'll speak a little bit more about that. But I wanted to make the point, and I think it's already been made by other presenters today, that we need to consider an expanded frame of reference with regard to sex effects in disease, not just with regard to incidence. That's one feature, yes, an important one. And so I choose type 1 diabetes here, which I just told you has relative parity in terms of numbers of male and female cases, to show you a couple of examples of that. So these are data on the risk to offspring of mothers who have type 1 diabetes. And on the left are some traces for mothers who presented with their disease before 10 years of age. This is a more common age at onset. The risk to their sons is far greater than the risk to their daughters. But if those mothers presented with type 1 diabetes after 10 years of age, there's relative equality in terms of the risk. So obviously children of type 1 diabetic mothers have higher relative risk versus the general population. But I think this is a very interesting pattern that suggests probably some epigenetic patterning that's coming from mother to child. So it's likely that the future of the risk to the offspring is associated with the onset of diabetes in the mom relative to her time at puberty. There's also evidence that there are sex differences with regard to presentation with islet autoantibodies. This is by far the best, most robust biomarker that we have for type 1 diabetes risk before actual presentation of the disease. There are a variety of different islet antigens that turn out to be targets for autoantibodies, and these are individuals that are separated by age, youngsters under 4 years of age, and this group here presenting from 15 to 20 years of age. There's quite a strong difference with male cases having higher titers of these antibodies than females. So I just use these as one of probably many, many examples from all of your areas of research that there are more features here that we need to understand with regard to sex differences than solely whether or not a disease presents more frequently in one sex than the other. So with regard to this issue, and I think this will be a topic of really a whole session later on today, and already touched upon, we really don't know the relative contributions of different mechanisms to these effects with regard to autoimmune disease. Clearly there are effects of hormones, and I'll show you some evidence from our group that supports that idea. But there are likely also very important contributions from X and Y chromosome-linked genes, whose behaviors can be epigenetically patterned, particularly the X chromosome. There are undoubtedly differences between males and females with regard to responses to environment, and the prior session was solely predicated on this idea, largely both with regard to vaccines, but also with regard to brain development. and an area that I think is really deservedly garnering a huge amount of interest, which are sex differences in epigenetic patterning and regulation of genes that have nothing whatsoever to do with sexually differentiated tissues. So these are data from tissues like liver and kidney, where there are clearly differences in the expression of many, many genes based upon the sex, for example, in inbred male and female animals. So we have very limited understanding of these mechanisms, and sadly, I think, perhaps tragically, no therapeutic interventions that are predicated on understanding these mechanisms. So the other huge feature that is impacting the frequencies of immune-mediated disease now is really something that we've seen over the past 50 or 60 years. These are aggregate data on the dramatic and welcome, of course, decline in pathogenic infectious diseases in children since the end of the Second World War, rheumatic fever, tuberculosis, and so forth. And this, of course, reflects differences in a variety of different aspects of public health. And concordant with the declines in those infectious diseases of childhood, we've seen a dramatic rise in the frequency of many of these immune-mediated diseases, including type 1 diabetes and MS that I already touched on, but also Crohn's disease. We have a large clinic at the Hospital for Sick Children that is seeing kids with Crohn's disease at 6 months and 2 years and 3 years of age. These are very, very serious diseases when they present and cause massive failure to thrive. And also the increases in asthma, ectopic dermatitis, you name it, the list goes on and on. So these are simply concordances in time. They're not telling us anything about cause and effect. But I think the graph on the left is showing us what our immune systems were designed to do. And this is what were evolved to do. This is what our grandparents and great-grandparents survived because their immune systems were able to survive these infectious insults, and we're no longer confronting those as frequently early in life. So clearly there have been dramatic changes in public health in developed nations and increasingly in developing nations with regard to water supplies, treatment of milk, products with pasteurization, the way that we perform husbandry of animals that we consume, of course, are increasingly deplorable diets, and xenobiotics, drugs, in particular probably antibiotics with regard to my comments today. And one of the sort of harmonizing notions is that these kinds of changes have been associated with a dramatic change in the composition of the microbial universe with which we come in contact. And of course, we have microbes living on all of our mucosal surfaces, but the highest concentration by far occupy our guts. So you've got about 2 pounds of bacteria living in your intestines right now, and you should love them because they're taking care of you as well. So the sex differences in the responses to environment that I just mentioned, and also in the way that genes are interpreted and played within different tissues, and the impact of hormones have all been topics that are impacting on animal models of autoimmunity. And since that was the focus of this session, I thought I would focus my comments there. So I've done quite a bit of both Google stalking and PubMed mesh term analysis to try to find papers on several of these common autoimmune disease models that actually use both sexes, or even mention which sex of animals they use. You'd be surprised how many don't even list them, and apparently reviewers and editors don't ask. There are a couple of very well-used models that mimic some aspects of multiple sclerosis, these experimental allergic encephalomyelitis models in a couple of different strains of mice. There is different pathology between sexes in one of the strains, relative similarity in the pathology between males and females in another strain, but the number of studies that use both sexes, almost impossible to find. There are a couple of groups that have been really dogged about doing this, and I'm sure they're having to argue all the way with their reviewers, but I really could only find two groups worldwide that are doing that for EAE. For systemic lupus, which can be modeled in a couple of different strains, the impact of the disease pathology in the mouse is far greater on the females than the males, and I couldn't find a single paper that actually uses males, and I understand why. You have to grow many more of them in order to see phenotype effects, but that would depend on which phenotypes you were looking for. If you're only looking at onset of disease, of course you don't see it. So that is back to my earlier question or issue about trying to think of this in a more layered, multidimensional way than just a presence-absence phenotype. There is a model of autoimmune diabetes on which my group and many others have worked called the non-obese diabetic mouse. This displays a sex bias of about 2 to 1 of females to males. And the number of groups that have looked at both sexes are relatively rare. And that's unfortunate for them because for us, having studied both sexes has turned out to be an incredible amount of fun. And we found a lot of things that we weren't actually looking for, which in my life has always been the best science. In terms of causal factors for type 1 diabetes, we also work extensively in human studies, but I won't speak of those today. There are a number of similarities between this NOD mouse model and humans, some similarities in genetics, a very clear T-cell lymphocyte-mediated response against the beta cells in the pancreas. And I had mentioned those islet-specific autoantibodies. We see them in both the NOD model and in humans. I mentioned that the incidence is greater in female than male NOD mice. The onset here is in adulthood, so we have to wait for a while. But as I had also alluded to, the incidence of type 1 diabetes in humans is relatively equal, and the onset in early childhood to adolescence. And in fact, the age at onset is dramatically declining. The greatest excess of new cases is in kids under 6 years of age. So this is very much an early onset disease. So a number of years ago, we were studying some of these aspects of sex under a standard laboratory environment of specific pathogen-free colonization. This is diabetes incidence over weeks, the males in blue and females in red. And to do a completely different set of experiments, we re-derived the animals into a germ-free environment in these isolators. And the sex effect that we and many others had reported for years went away. So that suggested that the sex of the host was somehow impacting on the microbes to which these animals are exposed or harboring, and that was having a strong effect on type 1 diabetes risk. So we've studied this in some depth, and I'll just summarize a couple of features. So Colin mentioned the microbiome, an area that we've worked on. One can use some high throughput DNA sequencing methodologies to ascertain the composition of organisms in a community. Increasingly, we're trying to actually understand what they do rather than just take a census of what they might be. And so we applied this kind of sequencing approach to the microbes in the guts of these NOD mice, these inbred mice, at the age of weaning, at puberty, and in adulthood. And the Venn diagrams here are meant to point out that at weaning, we can't discern a difference in the composition of the microbes living in the guts of the males and the females. But as they progress through puberty and into adulthood, we begin to see a clear delineation of difference in the composition of these organisms. So because it's a mouse model, one can do cause and effect relationship studies, and we were able to orally gavage groups of young females, either with adult male or adult female gut microbes, and change the composition of the recipient's microbiome over a persistent period of time. And the nature of the change that was provoked by the microbiome transfer was dependent on the sex of the donor. So somehow the sex of the donor provided compositions of the microbiome that actually persisted in the females. The reason that we were so interested in these findings and continue to work on this is that in contrast to unmanipulated females shown in the black, females that received these male microbes as weanlings shown in blue were dramatically protected from type 1 diabetes. But if they had been provided with adult female microbes at that same young age, shown in the red trace, they weren't protected. So this certainly suggested a strong cause and effect relationship, and we're still very much trying to understand how this works. And given the brevity of the comments today, I won't tell you too much more about our work there. I'll mention one last piece of information that might be of interest to this group, which is that the females who had received those male microbes at weaning actually displayed higher levels of testosterone in their serum, and in fact a number of precursors of testosterone. These are females on the left in green that had not been manipulated, and in the blue box and whisker plots are females that had received the male microbes. This is either 3 weeks later or 10 weeks later, and so their testosterone levels were elevated. So at least one of the feedback components that is instructive for testosterone levels, at least in males, appears to be mediated by the microbes that are growing in their guts. So that was another question that we had not set out to ask, but are glad that we bumped into. So the mouse models are excellent for trying to dissect these features, as all of the speakers have mentioned today. And we're also working in longitudinal studies with very young babies who have genetic risk markers for type 1 diabetes to try to ascertain features of their microbiome and their immune responses to those microbes that may be instructive about progression of this disease. So thank you very much. Do I have time for a question?
Thank you. That was a
Okay.
nice
Yes, go
overview of the sex differences in autoimmunity, and I'm a rheumatologist, so you were speaking my language. However, I have a question, and that is, have you or anyone else in the room done an NZB, NZW mouse for lupus? Because the thing is
Sorry, done what? With an
with a mouse model of lupus.
I've worked a little bit with.
Yeah, well, I actually don't study lupus, but I study some other diseases in mice, and we can't get the disease. If we have one sex, like males, and then we do the same protocol in female mice, we don't get the same outcome. So the
This
question,
is an NZB, NZWF one?
I use
Oh,
that, sorry, I'm very
okay.
sorry. I use that as an example.
Okay.
I study mice. I study a model in male mice. I can't, if I do the same protocol
I see. Yeah.
in female mice, even manipulating it a bit, I don't get the same outcome, the same incidence of disease, the same severity of organ involvement. Based on all your background, vast experience, are you willing to say that that can mostly be explained by the microbiome?
No, absolutely not, because you haven't done any experiments yet. But we could talk about some things you could try that wouldn't be
Well,
too taxing, that
I
might give you an idea.
No, I try to get grants, so I do that. But I'm just curious where, we have to push the models.
Yep.
Much more than the great insights we've had today. Yes, they're different. But I think that as you build the models, we need to think microbiome may have a part of it, but it's probably much more complex.
Absolutely.
Or not, than we know.
Absolutely. So we'll talk. Yes, hi.
Just a brief talk. This was a great presentation. Since you gave a very complete delineation of potential causes, what do you think about the role of microchimerism in explaining some of the sex-dependent outcomes in immunity?
That's one of the sort of classic experiments, right, that really started immunology, was microchimerism in cattle, right? And it's striking, as you mentioned that to me, I'm thinking to myself, when's the last time I had a conversation on this topic? I can't think of any evidence people have brought forward, certainly in animal models, for this. But as was just described, placentation is a very different process. We may be working in the wrong model altogether, and it might be more useful to be working in pigs or something that is more similar to human placentation. I can't think of any data. Totally scum. Oh, somebody's got it. Who said they did? They have data. Oh, you can't either. Yeah. So more to do. Thank you.
Okay, if we could welcome Brian Neiman, also from Sick Kids, for his talk on sex and mouse brain anatomy during health and treatment.
Super, thank you. And I'm coming from a bit of a different approach. So we don't do a lot of work in my lab looking at sex differences, but we've certainly tried to keep aware of opportunities to see when sex differences may be important. And I'd like to tell you today really mostly about one case, and I'll focus on that almost exclusively, where we found that sex may be a very important mediator in the outcome of a particular condition. And the variable we're going to use as an output is anatomy. And I'll apologize, I too am guilty of using some inappropriate language maybe. So at least in my case, we are really talking about geometry. So at that level there is, but it's more of a difference in size, not really in form. So I'm going to start just by talking about sex differences in the brain in healthy mice, looking at males versus females, and a very interesting model looking at how those can be explained by either hormones or chromosomes in a 4-core genotypes model. And then I'll focus in on the particular case that I was alluding to, where we're looking at how cancer treatment affects brain development in children. In particular, in the example I'm going to talk about cranial radiation and how that is dependent on sex, and in particular if we manipulate the immune response, how sex modulates that outcome. So how do we measure anatomy? So primarily the results I'm going to talk to you today are based on magnetic resonance imaging, which of course is a very common way of looking at the human brain. We use it also to look at the mouse brain, and I'm showing an example here of an individual mouse, in this case an image that was acquired ex vivo. And the nice thing about doing MRI in the mouse is you get a whole organ perspective. So you get the whole brain from the cerebellum right up to the olfactory bulb here. And you can see in this example the white matter comes out dark, and you get a very nice distinction between white matter structures and gray matter structures, and you can delineate many of the regions of anatomy that we would like in distinguishing impact of a particular pathology on the brain. There's a bit of a challenge in moving from the human to the mouse in doing MRI, and one of the challenges is it tends to be a little bit less efficient, and the imaging takes longer. So one of the ways we've addressed that is developing a really high throughput strategy. We actually image, in this case, 16 mice at a time, and this can be done then in a way that enables studies that are quite large that we wouldn't really undertake otherwise. The other advantage of doing things this way is the analysis can be entirely automated by computer. And I'll just give you a little bit of an overview of how we do that, because we're going to be looking at a lot of maps showing anatomical differences in this way. So if we take a hypothetical experiment where we've got two groups, and each of these groups may have 10 or 20 mice in them, and we want to be able to compare the anatomy, what we do is we morph these groups of mice together so that the images come together to form what we call a consensus average. And this really means we've taken all of the individual points in each mouse image, and we've morphed them so that I can point at one spot in this consensus average and then track that backwards. So I've mapped that same voxel in individual images that make up the average. So I've just mapped those brains on top of one another, and I've had to do some stretching or pushing in order to do that. The advantage of doing that is then the differences in shape is encoded in the amount of stretching or the amount of shrinking that I've had to do in order to make that alignment happen. So the data that we're using as an output really is the intermediate steps here that force the alignment of the images onto one another. And so if I can look at an individual point like this one and ask, what does the volume change look like at that point as a result of this transformation, I can map that in more of a traditional plot, and then you can do a traditional statistical test at that point and ask, well, how are the brains different? In this case, I've got 5 mice in the one group and another in the others. I can then do that at every point in the brain, and if I map that over the whole brain, then I can get a grayscale image in the background here showing what the brain looks like on an MR image, and then I can overlay a color image that says these are the regions where I think there's a volumetric difference, and I can quantify that in this case as a fold difference, or later we'll do it as a % difference. So getting into our study then, so here's an example of doing that and taking those two groups to be males versus females. And I've shown this plot because it also shows two different ways of showing the data. So here's this color map type of version on the top, And then you can also then go through and segment out structures of interest and show the volumetric differences as a whole in those structures, and that's what I'm showing on the bottom. And so here's a plot showing in red. We've switched things around compared to Anne's talk, I guess. So red and pink, in this case, our boys are bigger, and more the cold colors in blue, our girls are bigger. And you can see some of the changes that you might expect. So the bed nucleus is larger in males, and we see that here. And we can also see the amygdala and some hypothalamic structures in the thalamus that come out as being bigger in males. So if we look at how this maps onto steroid versus chromosome dependence, we use the 4-core genotypes model, which I think has been alluded to a little bit today, where we've moved the SRY region of the Y chromosome, and we haven't done this, but we're using these mice, so that the autosome holds the SRY, and then you can inherit the Y chromosome separately from the SRY region, which is the sex-determining region of the Y chromosome, so that you can have female mice that now have a Y chromosome, and you can also have male mice that have XX chromosomes. So we can do the same type of map and then ask with these color maps, which are the regions that are dictated by steroid and which are the regions that in volume are dictated by chromosome complement. And so again, in red, we've got male mice, so these are male mice that have inherited the SRY region of the Y chromosome, and blue indicates that those mice are bigger, so we've got region in the superior colliculi and the periaqueductal gray, which we heard about quite a bit, as well as, sorry, I said that backwards, as well as regions of the amygdala and the bed nucleus. And then conversely, we can see regions where the chromosomes become more important. And interestingly, we see quite a different pattern. So the corpus callosum comes out there. And down here, the basal forebrain is more dependent on the chromosomes. So that's the backdrop in terms of what differences there are in the male to female brain. Now, we were looking at a study where really we wanted to know how does radiation treatment in treatment of childhood cancers affect the brain development. What we do see in kids that have been treated for cancer is as they get older, they have more problems with working memory, more problems with attention, and ultimately this affects their performance in school and ultimately their long-term quality of life and performance in the workplace. And so we want to, of course, be able to avoid those problems, but still be quite successful at treating the cancer. And this slide really shows that we are much more successful at treating childhood cancers than we used to be, and perhaps more successful than we are at treating equivalent adult cancers. The most common childhood cancer is leukemia, which accounts for 1/3 of childhood cancers, and we're able to cure those children over 90% of the time at this point. Another common childhood cancer are tumors of the central nervous system, medulloblastoma being the most common one, and I'm showing that on the bottom where a tumor's been surgically resected as indicated by the arrow. And again, we're able to cure, in the case of medulloblastoma, 80% of the children with medulloblastoma. So we wanted to develop a mouse model looking at how cranial radiation affects development. And the parameters really, we're looking at this lower bullet point. So half of survivors experience late effects, and these appear quite a while after treatment, and we want to be able to address those. So the mouse and the human show very similar phenotypes after we've irradiated them. This is looking at just total white matter volume in the human and in the mouse, and looking at how that varies as a function of time at a radiation treatment. So if you look at younger children in the green versus a little bit older children, they show a smaller white matter volume than control children who haven't received radiation, versus the mice which have a similar pattern where the times don't necessarily line up exactly here, but we see that earlier in treatment you have a larger loss in white matter volume than you do later in treatment. This is what the timeline looks like for the experiment I'm going to talk about next, where we're really looking at radiation treatment at an early age, and then following that through to early adulthood and asking how does volume change over this time course. This is now in vivo imaging, of course, and this is what the controls look like on the top and the irradiation-treated mice on the bottom. So we do that same kind of color map here. I'm showing it mapping just the whole structure and looking at regions where there's a change at that last time point. And we see quite large changes in brain volume, notably in the olfactory bulbs, in the dentate gyrus of the hippocampus, some other white matter regions where we're getting larger than 10% volume changes as a result of radiation treatment. But really the whole brain seems to be quite affected. Smaller levels, the whole brain really is showing differences in volume. None of the regions that we look at really show a volume increase, but there's significant volume decreases everywhere. So how does sex play into that? So initially we looked at just those results, looking at males versus females. We looked over the whole brain. There really wasn't a significant sex effect in volume over most of the brain. The two regions that did show a small sex effects were in the fornix and the mammillary bodies. And the sex effect really was in the growth after treatment. So in this case, the males show a slightly steeper rate of growth, and in this case, the females really don't seem to show any growth at all, whereas the males show some recovery. But these really seem to be small effects compared to the size of volume loss that occurred early. So our initial impression was that, yes, there's a sex effect in these volume changes, but it's probably fairly minor compared to the size of the effect itself. But we wanted to move on, and we've been looking at how neurogenesis, how radiation-induced apoptosis, and how inflammation play into this phenotype after radiation treatment. And so looking at inflammation, we wanted to look at how recruitment of peripheral monocytes into the brain plays into the long-term development after radiation treatment. And so to do that, we wanted to look at mice that had a knockout of this chemokine CC motif ligand 2, CCL2, which is involved in recruitment of monocytes into the brain, and it was motivated really by some other studies that have already been done showing that knockout of CCR2, the receptor for that ligand, results in improved cognitive outcomes. And so we had three groups in this experiment, wild type, heterozygous, and knockout mice, and we wanted to look at how that changed. So I'm just going to show one graph here as a summary, and then I'll map it onto the whole mouse brain. And this is now first looking in the males. So I've taken here the cerebellar white matter as an example, and here's the time courses of them. I realize they're a little bit hard to look at because there's a lot of points quite close together, but looking at the last time point here on the right side kind of gives the summary of what I'd like to work with in terms of talking through this slide. The wild type mice, as expected, after radiation treatment show quite a large volume loss. If we look at the heterozygous and the homozygous mice, we see that the homozygous mice in particular seem to do a little bit better than the wild-type mice. And I'm going to quantify this by talking about the difference between the homozygous mice, untreated, sham-treated, and radiation-treated, versus the difference between the wild-type sham-treated and radiation-treated, and I'm really going to normalize that by the size of the difference here. And so the plot's going to look a little bit funny in terms of the influence of that denominator, but what I really want to look at is the direction of the change. So if we do that, pink colors in this case mean that we've got volume recovery as a result of the knockout of CCL2. This is looking only at the homozygotes. And blue colors would indicate that we've got a worse outcome as a result of the knockout. And so we see the take-home message from this slide is really we've got widespread pink over the whole brain, many brain regions, and it's true over most of that time course that we were talking about, right at day 23, a week after radiation treatment, all the way up to early adulthood. So we might say, well, this is a great outcome. Inflammation is clearly involved in how the brain develops after radiation treatment in males. We look at the same pattern in females. This is looking at the same structure. And again, I'll draw your attention just to this right side. We look at the wild types. There's a difference as a result of radiation. In the females, in the homozygotes, there really is no benefit to knocking out CCL2. And in fact, the homozygotes actually seem to have a slightly larger growth in the sham-treated group, so that deviation is actually bigger now than it was in the radiation-treated wild-type group. So I can do that same map for females, and we get now almost all blue on this map. So we're getting a widespread, either no benefit or in fact possibly an impairment as a result of that knockout. And I'm just going to put those two together to see just the stark contrast in the color pattern as a result of that radiation treatment in the different genotypes. So to summarize, I'm just going to talk about here the sexual differences that we see in the brain are quite small, but and even at the level of the radiation treatment differences that we see, the sexual differences aren't that large. And so we might have stopped at that point and said, well, sex doesn't matter that much. But as soon as we start to manipulate that system, in particular manipulating the immune response, we see that the males and females, though they do have a similar radiation response, seem to get there by a slightly different way. In males, that recruitment of monocytes seems to be particularly important, whereas females, not so much. And so this knockout in CCL2 has a very different pattern in the two sexes. And so I'd like to thank my colleagues, Jason Lurch and Mark Palmert, who did the work in the 4 core genotypes that I talked about, and Jacqueline Gleave, Lisa, and Elizabeth that did the work in the radiation-treated mice. Thank you.
That is stunning. Thank you for putting that back up there. It's a great example of sort of the two sexes diverging in the opposite direction. So you're allowed to use that term, by the way. This is a clear sex divergence. But the recruitment of monocytes is your hypothesis that you've got. This is so broad through the whole brain. Did the radiation cause monocytes to come in and massively invade the entire brain? Or are you having some kind of a chemical substance that's diffusing throughout the brain to have this broad damaging or protective effect?
In the context of these mice, we think that we do get a fair amount of monocyte recruitment. Now, is it even through the entire brain, and does it map sort of completely uniformly? That's something we're still looking at. Obviously, in these mice, we're currently doing some histology to really get a handle on microglia, astrocyte activation. Is it different between the two sexes? And in terms of the recruitment that we're getting from the peripheral immune system, and that I don't have the answer on that yet. But certainly, it could be that there's differences in factors, but certainly knockout of CCL2 anyway, and recruitment of the mice with the receptor for that, or the cells with the receptor for that, seems to be an important difference between the males and females.
We have to come all the way to Washington to talk to each other because we're in separate buildings. That was great. You likely know that luckily these days we don't do cranial radiation very often on kids unless there's really a massive CNS invasion certainly with leukemias.
Not with leukemias, but with brain tumors.
Of course, but we do a lot of intrathecal methotrexate. Right? This is the sort of standard CNS-directed therapy. I don't know whether you have the imaging data on those kids, but I'm guessing that in terms of numbers of cases, there would probably be far more of them than the CRT cases for your medulloblastoma kids. So I'm just wondering if you've thought about if there's an opportunity to look in that setting where you're infusing an clearly anti-proliferative agent, and they get those treatments throughout a 2-year period. So.
Yes, we are working both with kids and with mouse models of chemotherapy treatment as well, and that includes systemic chemotherapy treatment as well. And that's the point we're up to, is really looking at the systemic chemotherapy agents, but obviously an intrathecal treatment with methotrexate in particular is something that we're interested in. The systemic treatment is also showing some sex effects that are interesting in brain anatomy to a lesser degree, but also in as part of that, we've been looking at heart function, and we are seeing with anthracyclines there's a very important difference, as would be expected from clinical observations in the mice that are treated with doxorubicin, and the heart function, which is more affected in females than in males.
Thanks very much. So the last talk of the session is a presentation selected from the poster collection to be an oral presentation by Alexander Kesselman, who's from Hopkins. Right? So you have a 10-minute talk.
All right. Thank you, everybody, for staying for the last talk. So I'll get right into it. Asthma is a chronic inflammatory obstruction of the airways, and it exhibits pretty severe sex differences, affecting mostly boys in childhood. And after puberty, that trend is switched, and females are dominantly affected in terms of incidence and severity. And so that's sort of why we're here. That begs the question of whether sex hormones and sex in general play a factor in disease development and progression in allergic asthma. So allergic asthma is a Th2 disorder, which means that upon insult with allergen, you get the secretion of cytokines like IL-4 and IL-13, and these cytokines then bind their cognate receptors on alveolar macrophages in the bronchoalveolar space. And through the action of IRS2 and STAT6 pathways, these cytokines elicit the polarization of these macrophages into an M2 phenotype, which then results in the secretion of a variety of factors, some of which that promote tissue remodeling, some of which that recruit T cells and enhance Th2 responses. They promote fibrosis, and other chemokines that recruit monocytes and eosinophils, et cetera. And these cells are very important in asthma. Their accumulation in humans correlates significantly with disease severity. And in the mouse models, where we can actually ablate these cells, we see sort of the diminishing of a lot of these characteristic features of asthma that we all know and love. So these alveolar macrophages are emerging as really important mediators of allergic inflammation in the lung. Virtually nothing is known about how these cells respond to either sex hormones or if there are sex differences in their responses to polarizing cytokines. Certain studies coming out of different macrophages, like bone marrow-derived macrophages or spleen macrophages, suggest that sex hormones like estrogen may promote an M2 polarization. But virtually nothing is known about this macrophage, and they're quite unique and tissue-specific. So I'm interested in addressing that question, and in order to essentially first start out by characterizing or asking if there are sex differences in macrophage polarization in the mouse model, I used an OVA model, which is an acute model of allergic lung inflammation. Alveolar macrophages are sort of initiators of allergic lung inflammation, so that's why we went with a chronic model. Now, no model is perfect. There are pretty profound differences in responses between genetic backgrounds and allergens, and we can have a conference just on that alone. But as you can see, this OVA model initiates a pretty robust Th2 type inflammation, resulting in mucus production in the airways, as well as eosinophilic infiltration into the bronchoalveolar lavage. So I then went on to ask whether the alveolar macrophages in these mice exhibit sex differences with respect to their polarization status. So in the lung, in the alveolar space, macrophages are actually quite unique. They express CD11C and cyclic F, which isn't typically found on the surface of macrophages. So using flow cytometry, we were able to sort of get these cells and assess their expression of a variety of polarizing factors. So YM1, which is a canonical mouse M2 marker, and INOS, which is a canonical mouse M1 marker. And as I hope you can appreciate, after challenge with OVA, both male and female mice had increased expression of YM1 on the alveolar macrophages, but females went above and beyond the males, and this is a representative histogram showing that. There is a sex difference with respect to M2 polarization in this model. INOS, which is an M1 marker, did not show any sex differences, suggesting that the M2 program is uniquely regulated in a sex-specific manner. So then we wanted to take these cells and ask whether some of these differences might be accounted for by differences in receptor expression. So do females express higher levels of IL-4 receptor or perhaps estrogen receptors, as we sort of hypothesized that estrogen is an important sex hormone driving these differences. So using flow cytometry analysis, I stained for IL-4 receptor alpha and gamma chain on the surface of alveolar macrophages for male and female mice. And as you can appreciate here, what we found was that OVA challenge elicited an increase in IL-4 receptor alpha expression on the surface of female mice, but failed to do so on the surface of male mice. Likewise, if you compare female macrophages to male macrophages with respect to estrogen receptor alpha expression, there is a difference there where females exhibit higher levels of ER alpha expression, actually reflecting a reduction in OVA-elicited ER alpha expression. So it's somewhat of a double hit potentially. So an increase in IL-4 receptor and a decrease in estrogen receptor alpha may account for some of these sex differences. So then I wanted to basically go to an in vitro model where we can control better for some of these things. And we took bone marrow-derived macrophages from male and female mice, and we asked simply if we stimulate these cells with IL-4, which is the canonical Th2 cytokine, do we get sex differences in terms of, or sex-specific levels of M2 gene expression? So the canonical 3 mouse markers that are expressed by M2 macrophages are YM1, FIS1, and arginase. And as I hope you can appreciate here, with increasing concentrations of IL-4, we see that 2 of these 3 genes, YM1 and arginase, are regulated in a sex-specific manner and are expressed in a female-dominant manner following IL-4 stimulation. And FIS1 is sort of in between, suggesting that some but not all of the M2 program might be regulated in a sex-dependent manner, which is quite interesting. So then I wanted to ask how estrogen or signaling through estrogen receptor alpha might impact M2 polarization in these cells. So when we pre-treat these cells with either estradiol or PPT, which is an estrogen receptor alpha specific agonist, we see that the expression of all of these genes goes up, and mostly when we treat them with PPT. So ligating ER alpha elicits higher expression of YM1, which is Chi3L3, retinol A, which is FIS1, and arginase, and more so in the females than in the males, interestingly enough. So the males weren't very responsive. And then to address the role of ER alpha in vivo, we actually went back to the mouse model and we repeated our FACS analysis on Lysam-Cree ER alpha floxed mice. So these are mice missing estrogen receptor alpha in their macrophages. And we found that both male and female mice, after challenge with OVA, showed a marked reduction in YM1 expression in the absence of a functional estrogen receptor alpha, suggesting that ER alpha is very important in promoting M2 polarization both in vivo and in vitro. Now to test the role of estrogen, we actually used an overectomy model. So these are mice that we overectomized at 3 weeks of age. We then sensitized them with OVA like we usually do, and then we implanted them with either placebo or estrogen-secreting pellets. And we did this intentionally after the sensitization period so that we don't alter T-cell responses, because T-cells are quite sensitive to sex hormones. So, and then we followed with our 2 aerosol challenges and took our mice down and looked at the M2 profile of the alveolar macrophages. And we found that in the absence of estrogen, so placebo-implanted mice failed to polarize to an M2 phenotype. If you reintroduce estrogen in these mice, you rescue that M2 polarization. And this was also reflected in eosinophil infiltration into the lungs. Suggesting that estrogen is quite important for these M2 responses in vivo, and likely that estrogen receptor alpha is also recognizing the hormone and contributing to this polarization. So one thing that we noticed with these mice is that when we took their bone marrow-derived macrophages, so these cells are cultured for up to 10 days in the absence of exogenous hormone. They're then polarized with IL-4. We actually see a somewhat of a long-lasting residual effect of estrogen implantation in these mice, whereby when we stimulate them with IL-4, we get higher expression of these M2 markers compared to placebo-implanted mice. So this actually made me think that perhaps there's some epigenetic regulation going on here, because estrogen receptors are known to hang out with a lot of histone-modifying factors. So we wanted to see if perhaps the promoters of these M2 genes are regulated in a sex-specific manner. So we used CHIP qPCR to measure histone acetylation and trimethylation of lysine 4, which are activating markers, as well as the silencing marker, which is lysine 27 trimethylation. And we found that these activating modifications were largely female dominant across these promoters. So, and then with respect to the silencing, this trimethylation of lysine 27, it was, there was a trend to male dominant response there. So suggesting that M2 promoters are actually regulated in a sex-specific manner, and there may be a role for estrogen at these, in affecting these modifications. And that's something I'm actively pursuing in the future. So to summarize, I've showed you that allergic female mice exhibit enhanced M2 polarization both in vivo with ova challenge and in vitro with IL-4 stimulation. That estrogen or PPT, which is an estrogen receptor alpha-specific agonist, enhances that IL-4-induced M2 gene expression. And either overectomy or estrogen receptor alpha knockout dramatically suppresses the M2 response in vivo to ova, suggesting that this may be at least one of the major ways that sex differences emerge in asthma and allergy. So this is basically our model. We are now trying to tease apart where estrogen receptor alpha may be acting in the cell. Estrogen receptors are actually very dynamic. They can influence signaling cascades. There's a protein-coupled receptor, GPER, that can elicit calcium mobilization and alter gene expression through those means, through non-genomic means. So we're actively trying to figure out at which which step in this program estrogen may sort of tweak the balance and enhance M2 polarization and increase allergic inflammation. But this is basically our model of what's happening in the females. And in the males, this pathway is at least diminished. And so we think that that may account for the sex differences that we see in adults. And I'd like to thank my PI, Nikki Heller, and our lab, and Johns Hopkins. And I'll be happy to take any questions.
Hi, Tom Curry, Kentucky. Very elegant talk. Really interesting, the role of estrogen regulating M2 promoters. So my question really for you and for the group is, we're looking at differences between male and female. And in the past, studies have used males because they're much simpler.
Right.
No snide comments from the group. But have you thought about the actual cycle of the female? This is why studies don't use females. Because if you're looking at estrogen regulation, there are high levels of estrogen during the proestrus phase of the rodent cycle.
Right.
So is this something you considered?
Yeah, absolutely. And two things to consider there. One is that the estrous cycle in mouse is actually fairly short, and if you have a protocol that spans several weeks, you're likely they're cycling through it. And then the overectomy model actually overrides that complication altogether. So that's sort of why we pursued that model as sort of, as a way to address that issue. Yep.
Really neat. Good series of models. So this M1, M2 polarization thing, so you probably hang with the immunologists enough to know that there's some amount of skepticism that in real life, in vivo, there are these extreme phenotypes. Asthma maybe is a little bit of an exception there because you have such a huge load, presumably, of your allergen antigen right in the lung. But my question to you was, do you know whether when you shut down the capacity for an M2-like polarization in response to your allergen, to ova, do you sensitize toward an M1, or are you really silencing macrophage activation altogether? What if you come in with a classic M1 sort of TH1 type stimulation?
That's a good question.
Do they present antigen in a normal way?
Actually, one bit of data that I didn't show is that in the ER alpha knockout mice, where we did see this dampening of the M1 induction, response to ova, the males actually went INOS positive. So they went toward an M2. It really depends on the cytokine environment, right? If you have sort of a competition between like interferon gamma and Th1 cytokines with Th2, if you have a mixed bag of responses there and you block, M2 polarization, you're going to go M1. But if it's sort of very Th2, you're probably just going to sit in an M0 type of phenotype if you're not going M2. Sorry? Which is what you're setting up in your model. Right. There are definitely different models. There's a triple allergen model that I'm pursuing now that's very M1, and we're actually interested in steroid-resistant asthma as well, which is sort of a mixed bag. It's very neutrophilic, so it's more of a Th17 type of response. So there's a lot of flexibility with mouse models, and that can be a problem because there's sort of, depending on what strains you use, depending on what allergen you use, you can get different results. But yeah, so I hope that answers your question.
Great.
Thank you.
So that was 4 really great talks, really interesting session, and we could thank our speakers for that. I'll turn it over to the MC who's got some more announcements.
Yeah, all right. So that kind of concludes the talks that we have set up for today. So it was really great, guys. I think there were some excellent talks. I'm sure there'll be a lot of great conversation for the rest of the day. There's a short break right now. 3:30 will be when we start the poster session, so feel free to hang up your posters if you haven't. If you want to get started a little early, that's up to you guys, but that'll run till about 5:30, and that'll end the day today. So we can start the break now. I would ask that all of our moderators, speakers, Dr. Clayton and Dr. Wilder, just to stay by. We're going to take a few pictures, and then we'll head out for the poster session. So thank you again to all of our speakers and our moderators for a great first day.